the first part of our roundtable, we would like to talk a little bit about the challenges that are common in this area or in this part of drug development workflow. So, Anne, from your perspective, what do you think is the main challenge for API development?
So I guess from where I'm working, we're talking mainly about particle size and particle shape and the challenges that those pose to the development and the production of APIs. So particle size and shape are critical to quality for APIs. And to start with, a big part of that is how it affects dissolution and bioavailability. But the particle size, its distribution and particle shape will also affect processability. So in part of that scale up and optimization process, because it affects how the powders will flow and how they pack together. So for instance, if you have finer particles, they'll have a higher surface area, they're likely to be more cohesive, more likely to clump together and making them flow poorly in the process, more likely to form bridges in hoppers and bins and things like that during processing. So particle size is key, so making sure you understand what impact the certain size will have on the processability. But if you go beyond that to look at particle shape, that will also affect these processes. If you have particles that are nice and close to circular, then they're much more predictable in the way that they flow. They'll flow more easily. Whereas if you have elongated or rough particles, then they tend to lock together and they won't flow as easily. So in terms of the challenges from particle size and particle shape, it hits APIs in a lot of different areas. So whether it's the function in terms of dissolution, but also in how you process them, how those powders are going to behave in the process.
Thank you, Anne. I think one maybe small follow-up question from me. So if you mentioned the particle size and size distribution, so if we talk about packing of the particles, wouldn't it be actually beneficial to have some differences in particle sizes so that you can pack them more efficiently? Or is this about something else than efficiency of packing?
Yeah, certainly you can. It depends what you want to achieve. If you want a high packing density, you probably do want to mix together fine and coarse particles so that you can effectively pack the small ones around the big ones. So yes, certainly packing density, you're often blending materials perhaps to achieve that packing density. Or maybe it's something that you look at further down the line when you're choosing what excipients to go in with the APIs, whether you want overlapping size distributions or how close together you want those to be. So yeah, it's certainly a consideration.
Thank you. So Kruti, you bring the knowledge of solid form characterization and structural analysis to this roundtable. So why would you say these are important?
Oh yeah, so let's talk about structural analysis. I think I always use this term for like umbrella term, right? So Anne just described, the macro appearance of your drug product or your excipient, whatever that it is, right? And that is, I call it a macro or, a micron size structural characterization or structural analysis. So what I bring into on the table now is what is the structure, right, of your API? What is the packing of your API? So now here the packing is a little bit different compared to what Anne was saying. The packing is how do they crystallize, right? Whether or not they form crystal or not, right? Or sometimes, our customers, they just don't like their API to be crystal. They want a amorphous API. So I think that will be the two things we have to think about even before we, talk about structural analysis, right? One is like whether or not you want your API to be a crystal form or an amorphous form, right? So if you figure out which route you want to go, then you have different strategy to do that, right? If you are sure you wanted an amorphous form, then what we're trying to do is we're trying to figure out whether or not your amorphous form keep like that, right? As a function of time, as a function of your shelf life. Or if you're going to go to the crystal form, then whether or not you can find the most stable one and whether that most stable form is going to, be there on your shelf. Basically, that's kind of the same idea, but using a different way, So I believe that's the two questions that have been asked most of the time when I'm on customer side, when I do the sales call, when I do something, like that.
I see. Thank you very much. And then, Lieven, what about you? How do you see this? Are there any further challenges you think there are in the API development process?
Yes, from my side, I look at elemental composition, and when for the development of APIs, or during the development of APIs, you can also introduce some impurities. So purity of API, of compounds, is also a challenge to analyze them, and also making sure that the impurities are at a very low level, because we all take medicines, drugs. They want to cure you, and not that they make you sick because of lead concentration in the pills is on the high side. So purity is an important aspect as well for API development.
And maybe then a short follow-up question. So what kind of impurities do you mean specifically? Because Koji has also mentioned that there can be a mix. So he was talking about crystalline and amorphous phases. So is this can you address the same question using elemental analysis, or is this something else that you mean by purity?
It's something else. So the purity is here, I'm talking about the elemental impurity of, let's say, elements that can be introduced during the development of using the vessels where the product is in, where some chromium, manganese, copper as elements can be on the high side. But can also be starting from the raw material, that there is a low concentration of arsenic, cadmium, lead, mercury, all of those elements you don't want to have, but it's common at low concentrations. And then is the question, is the concentration low enough? But another one can also be the catalyst residues. During the development process, catalyst introduced, but you don't want to have that in your end product. So then you also check for rhodium, platinum, palladium, those kind of catalyst elements.
So this is really about some things that shouldn't have been there or should not be left in the reaction mixture as you process.